5 layers. The outer surface is lined by stratified, non-keratinized squamous epithelium about 6 cells thick. This corneal epithelium rests on a thin basal lamina supported by a thick specialized layer of corneal stroma known as the Bowman's membrane. The Bowman's membrane is particularly prominent in humans, which is a distinctive layer of collagen fibers about 7 to 12 micrometers thick. And then we have the bulk of the cornea, which is the substantia propria or stroma. This consists of a highly regular form of dense collagenous tissue forming a thin lamellae. So within the stroma, fibroblasts can be seen. These cells have elongated nuclei and barely visible cytoplasm. We refer to the fibroblasts as keratocytes. These keratocytes are scattered in the ground substance between the lamellae. The inner surface of the cornea is lined by a layer of flattened endothelial cells, which are supported by a very thick basement membrane we call as the Descemet's membrane. The corneal endothelium is made of simple squamous epithelium covering the posterior surface exposed to the aqueous humor. So the 5 layers of the cornea are the following. We have the corneal epithelium, the Bowman's membrane, the substantia propria, and then the Descemet's membrane and endothelium. Now let's go back to the retina. So the retina is the posterior surface of the eye responsible for photoreception. It is composed of 10 layers. So this will be from the outer layer to the inner layer. First, we have the pigmented epithelium. So it is made of simple epithelium forming the blood-retinal barrier. A photoreceptive layer of rods and cones, which are membrane discs of the rod and cone neurons. Then we have the external limiting membrane, which is a layer of tight junctions between rods or cones and other neurons. Next, we have the outer nuclear layer, which is now composed of the nuclei of rods and cones, the outer plexiform layer, which are formed by synapses, the inner nuclear layer, where we see the nuclei of different neurons, then the inner plexiform layer, where synapses are also seen, the ganglion cell layer, where cell bodies of bipolar neurons are seen, the optic nerve fiber layer, where axons of ganglion cells are seen that project into the brain, and lastly, the inner limiting membrane or the basement membrane. So when you look at your specimen later on, you look for the retina. Okay, so this is how it would look like. So ideally, ganito siya. Where is the pigmented epithelium? There, number 1. Number 2 would be the photoreceptive layer of rods and cones. Number 3 would be the external limiting membrane. Then we have the outer nuclear layer, the outer plexiform layer, the inner nuclear layer, inner plexiform layer, the ganglion cell layer, optic nerve fiber layer, and the inner limiting membrane. Once you have located all 10 layers, scan through the entire retina to look for the fovea. So the fovea is a small depression in the retina that lies at the center of a larger oval-shaped area we call as the macula lutea. The fovea is on the visual axis of the eye and therefore provides the area of highest visual acuity or resolution in the retina. This fovea consists entirely of cones. And then the center of the fovea is called the fovula. Take note that in the retina, we have blind spots, or we have what we call the non-visual spots and visual spots. So the fovea is one of the visual spots. Then we have the cochlea.
Ears.
So the cochlea is a spiral canal shaped like a snail shell. We find the cochlea in our ear. So take note that the cone-shaped mass of bone surrounded by these turns of the cochlea is called the modulus. So this contains a canal through which fibers of the cochlear nerve pass. A mass of neurons belonging to the spiral ganglion lies to the inner side of each turn of the cochlea. The parts to be identified in each turn of the cochlea are the following. We have the scala or scala vestibuli, which is the upper compartment, the scala media or the cochlear duct, the scala tympani or the lower compartment. The cochlear duct is filled with endolymph. Perilymph fills the other two scalae. The vestibular membrane, also known as Reissner's membrane, marks the roof of the cochlear duct and consists of two layers of simple squamous epithelium. This separates the cochlear duct from the scala vestibuli. And then a thicker basilar membrane forms the floor of the cochlear duct and separates it from the scala tympani. The organ of Corti is a highly specialized epithelium consisting of hair cells and supporting cells. So the organ of Corti is located in the scala media. Then we have the olfactory epithelium. Take note of the different parts. We have the anterior cranial fossa, the paranasal sinuses, turbinate bones, nasal cavity, nasal septum, and orbital cavity. Now, where should you look for the epithelium or the olfactory epithelium? You're going to look for the olfactory epithelium on this part, dito sa may nasal cavity. How would it look like? Okay, so it looks like this. Take note of the following structures. The epithelium is pseudostratified. It is much thicker than the epithelium lining the respiratory mucosa. And within the epithelium, there is a superficial zone of clear cytoplasm. Below the epithelium, we find several rows of nuclei. Or in the epithelium, we have the lining cells. Okay? And using special methods, 3 types of cells can be recognized in the epithelium. We have the sustentacular cells, the olfactory cells, and the basal cells. Take note that the nuclei of the sustentacular cells tend to be in the top portion of the layer. The nuclei of the olfactory receptor cells would be found in the middle portion, and the nuclei of the basal epithelial cells tend to be in the bottom portion. In the lamina propria of the olfactory, nasal cavity, okay, we find the lamina propria, where we find the Bowman's glands, nerves, some veins, as well as capillaries. Then we have the taste buds. So our taste buds are the chemoreceptors for the sense of taste. So they are mainly located in the epithelium of the circumvallate papillae of the tongue, although they are also found scattered in other parts of our tongue, palate, pharynx, and epiglottis. So the taste bud is a barrel-shaped organ occupying the full thickness of the epithelium and opening at the surface via the taste pore. Each taste bud contains about 50 long spindle-shaped cells which extend from the basement membrane to the taste pore. Classically, two types of cells are described in the taste bud: the light gustatory cells and the dark supporting or sustentacular cells. A third cell type, which is the basal cell, is now generally recognized and may constitute the precursor of one or both of the other cell types. Both gustatory and sustentacular cells have long microvilli extending into the taste pore. The microvilli would contain a glycoprotein substance thought to be secreted by the sustentacular cells. Okay, that's it for our special senses. So when we see each other after the midterm exam, you will be able now to check or have the chance to check the slides of our special senses before you have your performance task. Okay, thank you and have a nice day. Okay, so do you have any questions or clarifications? Okay, so there's none. I will hand it over to Sir Ishmael. Okay, so thank you.
Okay, so now I will be giving a pre-lab discussion on the two exercises, exercise 10, which will be the last exercise for the midterms, and exercise 11 for the first exercise for your finals. Now, sorry, I can't turn on my camera. I don't have a I don't have a camera. Um, so kindly listen to my disembodied voice as you look at the screen as I present my slides. So blood is our exercise 10. It is a specialized connective tissue. In older references, they used to refer to it as liquid connective tissue. Some other more recent references, they call it a fluid connective tissue. But regardless, it is classified as a connective tissue, and blood is where cells are suspended in fluid known as plasma. Your blood is circulated around the body by rhythmic contractions of the heart. 5 liters of blood in an average adult moves unidirectionally within the closed circulatory system. When we say closed, as you can see here, anything, let's say here, the blood coming from the lungs, going back to the heart, setting to the arteries, going to the capillaries above and below the body, all moving back through the superior and inferior vena cava, back to the heart, and then to be oxygenated into the lungs, and then back again to the heart. Closed siya. Hindi siya, your blood, you're not leaking blood. Okay? It's all going to come back in this closed system. Unless you're bleeding, I guess. So, so for histology, now blood is not a primary tissue sample under histology. It is more on the purview of hematology. But we'll just go over a little bit of the formed elements, your blood cells, which we will be observing under the microscope for our activity. You have your red blood cells or your erythrocytes, your leukocytes, which are your white blood cells, and your thrombocytes, your platelets. As a review, so blood, this topic is not new to you. You have this has always been discussed in your previous subjects from ANAFI to PMLS. You already know, you have at least a general idea of what the blood is, what it does, how important it is in the body. To just cite a few of the functions, it distributes nutrients, it regulates body temperature, it maintains acid base and osmotic balance, and it carries and transport a plethora of varying substances like oxygen, carbon dioxide, or gases, metabolites like nutrients, hormones, and other molecules. Oh, and it's also involved in our immune defenses. Now, blood, you already know its composition. You have your plasma, which is 55% of whole blood. Your erythrocytes, your red blood cells, is 44% of whole blood. And your buffy coat, this small sliver here in between the two, is less than 1% of blood, and it contains your platelets, leukocyte, and your leukocytes. In your leukocytes, there is a percentage of you have a percentage of white blood cells are your neutrophils, so 50 to 70%, the largest portion. Lymphocytes are 20 to 40%, monocytes 2 to 8%, eosinophils is 1 to 4%, and basophils have less than 1%. So plasma, you if we separate your whole blood via centrifuge, your formed elements will be formed here from the buffy coat to the red blood cells, while on top would be the fluid portion of your blood, which is your plasma. In your plasma, it also contains salts, minerals, your sodium, potassium, calcium, chloride, your water, and also your proteins like your fibrinogen, albumin. As such, these five, those are your plasma. Your buffy coat, it contains your white blood cells and your platelets. It composes less than 1% of the total volume of the blood sample. And it's called the buffy coat because it is usually buff in hue. I had actually had to look up why they call it buffy coat. Ayun, hindi kasi si Buffy. Okay, that was a bad joke. Moving on. And then lastly, your red blood cells, 44% of whole blood, and is referred to as hematocrit in an anticoagulated whole blood centrifuged sample. So let's talk about your formed elements. We'll start with uh, the most of them. This is your erythrocytes. Your erythrocytes, these are terminally differentiated cells. When we say terminally differentiated, it means they are so well developed that they lose their ability to proliferate. So, so that's what they are. They're very well. They are very developed into a certain function that they do not accomplish any other functions anymore. And their function is, of course, transporting gases, specifically your oxygen and your carbon dioxide, to and from the lungs and into your tissues. So they contain this oxygen carrying protein, your hemoglobin. You have four heme groups, and each attached to a globin. And yeah, as a general characteristic, it's red. It lacks nuclei and organelles. It's mainly made up of hemoglobin as the main component. They uh, carry out oxygen and transports carbon dioxide via its specialized membrane. The amount of oxygen in blood, called the oxygen pressure, is highest in the arteries because they mostly carry oxygenated blood, and decreases in tissue capillaries where exchange of oxygen and carbon dioxide occurs between blood and tissues. To illustrate this, we have this graph. Here in the venous blood, oxygen pressure is very low because there's little remaining oxygen. But once it reaches into the lung capillaries, they get oxygenated. The carbon dioxide is also removed from the blood, so more space for the oxygen. Once it reaches back to the arterial blood, there's a slight decrease because it's no longer exposed to the mostly the oxygen pressure coming from the external
Yes.
environment in the lungs. And then once they distribute the oxygen back into the tissues and take up carbon dioxide again in the capillaries, it decreases until it has to return back to the veins and then into the heart to be reoxygenated. It has a general lifespan of 120 days, and its shape is this biconcave disc. A biconcave disc, what does this even mean? Biconcave meaning there's a concavity, meaning an indentation on both sides. That's why it's biconcave. There's one on this side, and then the other side of this red cell, there's another. So, ayan, one concave and another concave. Two concaves, meaning biconcave. And this biconcave shape, it helps increase in the surface area of the cell to allow more oxygen to be carried by your red blood cells. And also, It provides some flexibility to thanks to its shape. It has this characteristic called deformability, which allows passage to small vessels in the microvasculature while maintaining a constant surface area volume ratio. You can see here in this slide, this is a blood vessel, and passing through this blood vessel are red blood cells. They are stacked in what you may be already familiar, your Relu formation. They're stacked like this so that they can pass through very small capillaries without difficulty. And if you can already imagine, what would happen if suddenly this biconcave disc becomes a sphere, like in your spherocytes? This would end up making it difficult for blood to pass through these capillaries, decreasing the amount of oxygen being facilitated to the tissues, decreasing the oxygenation, making people more, the people with the spherocytosis, you know, a bit a bit more winded down because they don't have as much oxygen as a normal person. And that's your erythrocytes. For your leukocytes, your white blood cells, they leave the blood and migrate to tissues. Unlike your red blood cells, they don't leave the blood vessels normally. Diapedesis does occur where they just follow along, but they aren't supposed to leave the blood vessels. Your leukocytes, they can leave the blood and then migrate to tissues where they become functional and perform various activities related to immunity. There's two groups which we differentiate based on the presence or absence of specific granules, your granulocytes and your agranulocytes. The prefix a here meaning wala, none. So agranulocytes means there's no granules. Now, despite saying that there are no granules, both of them, however, contain non-specific granules. When we say non-specific, we're usually talking about your lysosomes. You know about your lysosomes already, right? They are organelles found in almost all of your cells, of course, in varying quantities. But your lysosomes, they also stain um, in some of the stains that we use for hematologic study. That's why we call them non-specific granules because they are seen in most cells. The differing factor is your specific granules, and your granulocytes they contain specific granules. Your neutrophils they contain granules that stain very faint or light pink. Or eosinophils they have granules that stain red or dark pink, and your basophils they stain dark blue or purple. So let's go over your granulocytes one by one. We'll start with our neutrophils. Neutrophils can be identified by their multilobulated nuclei, with lobules held together by very thin strands. So here, when we say multilobulated, this is one lobe, this is another lobe, this is another lobe, and this is another lobe. So four lobes, as you can see. This one has three. Oh, I guess you could see this is almost one, but regularly they have three to an average of four, mostly always four, but sometimes they can have three or five. Any more lobes, and perhaps there's a problem with your neutrophils. Now, because they have many lobes, because of this feature, we that's why they are also called polymorphonuclear leukocytes, or PMNs, or polymorphs. What does that word mean? Poly meaning many, morpho many shape, but in this case lobes, and nuclear. So many lobe nuclear, diba? Your leukocyte, your white blood, your neutrophils. That's why we call them your PMNs. Here's a slide of a patient with neutrophilia. Now, normally it wouldn't be this many neutrophils in your blood, but if you have a bacterial infection, it can represent under a hematological slide na makikita nyo may maraming neutrophils, as you can see in this picture here. The second type of granulocyte is your eosinophil. They're about the same size as neutrophils but have bilobed nuclei and more abundant coarse cytoplasmic granules. So let's take a look at that. You have here same size, your eosinophil and your neutrophil, same size. And they are bilobed, unlike the neutrophil here, which has three or almost four lobes. I think this is just a bar body, but your eosinophil has two lobes, one, two. But the most differentiating characteristic eosinophils have are these very dark red granules all throughout the cytoplasm. Can you see here? This is not the cytoplasm. These are the, well, it is, but what you see on top or within the cytoplasm are the eosinophilic specific granules that you can see here. That's why if you take a look at the picture on the lower right, the entire cytoplasm is filled with these granules. It's even on top of the nuclei, if you can see as well. Yan, maraming red na granules on your eosinophils. In patients with eosinophilia, this is seen in patients with helminthic infections and schistosomiasis. Um, what's more important, however, with eosinophilia is not the differential count. So if you can remember, what do we mean by differential count? Differential count is the percentage of white blood cells. Let's go back here. You have your neutrophils. It's normally 50 to 70%. Lymphocytes are needed 40%, monocytes to 8%, etc., etc. Your eosinophil, it's normally 1 to 4%. But even if it goes up to 10%, it might not be that abnormal if perhaps bumaba lang talaga yung neutrophils mo, or naubusan ka ng neutrophils, or naubusan ka ng lymphocytes, or both. Pwedeng mag-akyat ang eosinophil differential count. If this went up 10%, then that's not so big of a deal if your entire white blood cell count dropped. However, absolute count, what if instead of the regular amount, oh, I skipped, I skipped it, I went too far. The normal absolute count of your, and let me just look it up. I forgot already. So the normal absolute eosinophil count is 30 to 350 cells per MCL. But if in a high count where you get twice that usually, then that would be more clinically important compared to just saying uh, mataas yung or mababa yung differential count. Yeah. So absolute count is more important in eosinophils. Your basophils, the third of your granulocytes, they have naman, their differentiating characteristic is their dark purple granules. And as you can see here, this one and the pictures on the right are basophils. This one, so many granules, so many dark purple granules. This one also has dark purple granules, although what's happening here. And so it's same as this one as well. They also have two irregular lobes, but unfortunately, because of the presence of these granules covering the entire cytoplasm, it's hard to appreciate the shape of the nucleus kasi natatakpan siya ng granules. Like, look here, di ba? Or ito, you can somewhat see the granules, yung nuclei, I mean, then here, asan yung nuclei? Nakatago na siya, napatungan ng granules. Unfortunately for your basophils, they are less than 1% of the total leukocyte population. I have never seen a basophil in all my years of practice as a medtech, and probably neither will you. Now, agranulocytes, they do not have specific granules, but they do contain azurophilic granules with affinity for the basic azure A. The nucleus is spherical or indented but not lobulated. This group includes your lymphocytes and your monocytes. Your lymphocytes here, they lack specific granules, characteristic of granulocytes. They circulate in blood generally ranging in size of 6 to 15 microns in diameter. You have here red blood cell. Generally they're 5 to 6 microns naman. So your lymphocytes are the closest in size to your erythrocyte. So here they have another picture of an erythrocyte. It's almost the same size as the nucleus of the lymphocyte here. It's all basically 1.5 times larger than the than an erythrocyte. Your monocytes, however, they are much larger. As you can see here, there's an erythrocyte and here's a monocyte. You could say it's 2 to 3 times larger than a erythrocyte. And it's part of the mononuclear phagocyte system. Another key thing about your monocytes is they can also have this circular shape na nucleus. Uh, in that case, we'd have to differentiate it by manner of size. But most of them do have what we call this kidney bean shaped nucleus, or rather a C shaped nucleus, which allows us to better really identify it as a monocyte. Here's in the picture on the bottom right. You have a lymphocyte here and then a monocyte right next to it. The last of your formed elements is your platelets, your thrombocytes. Your thrombocytes, they originate from your megakaryocyte here. Megakaryocytes are typically not seen in peripheral blood. Um, they usually remain only in the bone marrow, and by the time they reach peripheral blood, they've they've separated na into these little tiny pieces called your platelets. So your platelets, they promote blood clotting and help minor tears or leaks in the walls of small blood vessels, preventing the loss of blood from the microvasculature. In stained blood smears, platelets can also appear in clumps. We call this your platelet satellitism, where they all gravitate to a point where they connect to each other. But yeah, yes. Your thrombocyte also has parts, but it was, of course, under light microscope. They're already so tiny. This is already HPO. Um, but they have parts like your granulomere, which is the darker zone with organelles, and then the hyalomere, which is a lighter stained peripheral zone. If you take a look at some of the platelets around, there's a darker zone in the middle, and then surrounding it is this faint pink hyalomere structure. In patients with too much platelets, we call this thrombocythemia. And here, masyado ng maraming platelets yan. So that's it for exercise 10. We now proceed to exercise 11, which is adjacent to our discussion, your blood vessels. So your exercise 11, as a reminder, this is again part of the finals, but we are covering it because of a incredible lack of time that we have. So we're gonna try to condense as much as possible to prepare you, and so that we can maximize our time in the laboratory. So I talk about the blood vessels, you need to know about the circulatory system. Baka hindi niyo pa na-discuss sa LEC, or kung na-discuss na sa LEC, that's great. But your circulatory system, it consists of the blood vascular system, which is just the all of the blood vessels, sans the heart and perhaps the lungs, and then your lymph vascular system, which is this green structure here. Your your blood vascular system or blood circulatory system, um, it's part of the cardiovascular system. But if when you say cardiovascular, of course you're including the heart. But without the heart, it's just your blood vascular system. It is, as we've described a while ago, it is a closed system. So blood coming from the heart, oxygenated blood, it's all going to the tissue capillaries, into your capillary beds. It exchanges gases and then becomes deoxygenated blood, returns to the veins, and then back into the heart. It will then pump these deoxygenated blood into the lungs, where they'll be oxygenated and go back to the heart. Your lymph vascular system is a network of drainage vessels for returning excess extravascular fluid, and it circulates lymph. The reason I am recalling this or re-emphasizing is because maraming estudyante sa ANAFI ang nakwento kasi nila ma'am na nagtuturo ng ANAFI na di nyo alam na separate ang lymph tsaka ang blood. Okay? So lymph is very different from your blood. Okay? And I'll discuss more on how that separates later. Now, for your blood vessel and also lymphatic vessels, they share the same structure of an inner, middle, and outer layer, starting with your tunica intima. When you say the tunica intima, it's over here, coming into contact with the lumen of your blood vessel. So it's coming into contact with blood. It is lined first with an endothelium. You can see here your squamous cells, a single layer of squamous cells here. And then in the middle, you have just more basement membrane and collagenous tissue. So that structure completes the tunica intima. In arteries, however, we have a internal elastic lamina, which I think you've seen with the elastic stain, although um, marami kasing elastic fibers doon. But yes, there is an internal elastic membrane here composed of elastin, which allows better diffusion of substances from the blood deeper into the wall. The middle layer naman of the blood vessel is your tunica media. It's predominantly smooth muscle tissue. In arteries, the media may also have an external elastic lamina naman, as you can see here, separating it from the outermost tunic. And the outermost tunic is your tunica externa or tunica adventitia. It is an outer supporting tissue layer. It consists of connective tissue alongside the vasa vasorum and nearby vasorum. What's the vasa vasorum? It's a blood vessel for your blood vessel. For your very large arteries, for example, or very large blood vessels, because they are so large, they need also their own blood supply. And your vasa vasorum allows oxygen and nutrients and waste to move from the lumen to the cells of the vessel wall. Um, Unlike here, if you take a look at this picture, rather than the blood trying to oxygenate this vessel from the lumen, there are blood vessels that extend into the tunica adventitia. That way, diretso na siya dito sa may musculature ng ating blood vessels na ma ano ma ma-supply ng ating blood with oxygen, nutrients, etc. So let's talk about your arteries. Your arteries, their job is to distribute blood from the heart to capillary beds of throughout the body. The cyclical pumping action of the heart produces a pulsatile blood flow in the arterial system. With each contraction of the ventricles, if you recall your ventricles, they're the chambers of the heart that pump, blood is forced into the arterial system, causing expansion of the arterial walls. Subsequent recoil of the arterial walls assist in the maintenance of arterial blood pressure between ventricular beats. This expansion and recoil is a function of elastic tissue within the walls of the artery. So if you have a rubber band around or a pingol, if you push it outwards, it recoils back to its original shape, yes? So yan, may elastic tissue rin kasi yung mga, some arteries, especially the ones nearest to the heart, which gives that sort of beating motion. Now, there are three types of arteries, ranging from your elastic artery, muscular artery, and then you have your arteriole. Your elastic arteries comprise the major distribution vessels and include the aorta, the brachiocephalic trunk, the common carotid and subclavian arteries, and most of the large pulmonary arterial vessels. Your elastic arteries, their characteristic shape is this is what you've seen stained by an elastin stain, your elastic arteries, if you can recall in exercise 4 in the connective tissue. It is this. This is an aorta here, class. And you can see there's pink thin stripes of these thin pink stripes here. These are your muscle tissue, smooth muscle. And then in between, which is these areas, these are your elastic fibers. So interspersed in alternating, you have muscle, then elastic fiber, then muscle, then elastic lamina again, over and over. So your elastic arteries are very heavily, maraming elastic fibers siya. An elastic artery is also known as a conducting artery because the large diameter of the lumen enables it to accept large volume of blood from the heart and conduct it to smaller branches. And it also contains vasa vasorum, as previously mentioned, in the tunica adventitia. The second type of artery is your muscular artery. They are the main distributing branches of the arterial tree, like the radial, femoral, coronary, and cerebral arteries. Their thick tunica media allows muscular arteries to play a leading role in vasoconstriction. In contrast, their decreased quantity of elastic fibers limit their ability to expand. So you might think na here, this is your elastic artery, and then here is your muscular artery, right? You might think that, oh, mas makapalang tunica media ng elastic artery compared sa muscular artery, but that's just because of the elastic fibers present in your elastic arteries. In your muscular arteries, there's more muscle here because there's more muscle here because it's not replaced with elastic fibers. Also, the tunica adventitia of your muscular arteries, it's bigger, and also yeah, it's bigger compared to the tunica adventitia of your uh, elastic arteries. Now, we mentioned that there is now a decreased quantity of elastic fibers. Fortunately, because the blood pressure has eased by the time it reaches these more distant vessels, elasticity has become less important. Your elastic arteries kasi, like your aorta, it is very near the heart. Meaning, so kung nagpa-pump yung heart mo, mas malakas yung pressure dun sa area na to. So, and of course, the nearer it is to the heart, the stronger the pressure. It's like if you have a faucet, right? If you have a faucet, if you place your hand so close enough to the to the exit point of the water, and then you turn the faucet on, mas malakas yung pressure, di ba? Ng tubig. Compare that if you place your hand much further away from the faucet, then there would be less pressure. Ergo, that's why your elastic arteries have more elastic fibers so that it can withstand that greater amount of pressure. And by the time the arteries have gone further down through the body, the pressure has eased, meaning mas konti na yung pressure, so it doesn't need as much elasticity compared to your elastic arteries. The third type of arteries are your arterioles. It goes even further away from the heart. These are the terminal branches of the arterial tree, which supply your capillary beds. They have the same structure. It also contains the three tunics, but because they're so small, they're much limited. So you have your tunica intima, there may red blood, may erythrocytes tayo sa gitna. This one layer of your tunica intima, one or two layers of smooth muscle around for your tunica media, and then your tunica adventitia is the rest. Your arterioles, they are critical in slowing down or resisting blood flow and thus cause a substantial drop in blood pressure. Because of this, you may see them referred to as resistance vessels. And you need the blood pressure to be lower by the time they reach the capillaries because you don't want naman na malakas like masyado malakas na pressure pressing against your organs. For example, you might end up starting to sweat blood at that point. So we just need a small, just enough pressure, but not too much. That's why we have your arterioles. After your arterioles, there's actually a fourth type of artery, but because they're much smaller, there's no light micrograph for them. You have your metarterioles. Your metarterioles, they are, if you have here your small artery, right, going down, it's the biggest one here. A branch is in arteriole, and then it branches even further into your meta arterioles. And your meta arterioles, that's the last step before we head to our capillaries. So you take a look at this picture here. You have your arteriole, right? We have an arteriovenous anastomosis, which is just a direct connection of from the artery to the vein. And from your capillaries, you have what you call your thoroughfare channel, which allows also connecting your arteriole to your venule. The most important aspect here is your capillary bed. So the capillary bed, this is where your exchange of blood and tissue fluid occur to exchange gases, to get waste products, and to deliver nutrients. And these capillaries, we also refer to them as the microcirculation. The microcirculation is the part of the circulatory system where the exchange of gases, fluids, and metabolic waste products occur. Exchange occurs mainly within the capillaries, extremely thin-walled vessels forming an interconnected network. Your capillaries, they will look like this. This is an artery, and this is a vein. Malilit talaga yung capillaries natin. So capillaries, they permit and regulate metabolic exchange between blood and surrounding tissues. They usually work in groups. We call them your capillary beds, whose size and overall shape conform to that of the structure applied. Like here, this is a muscle tissue and it's surrounded by your dark lines of capillaries, to which is conforming to the shape of your muscle tissue. Your capillaries, there are three types. You have your continuous capillary, your fenestrated capillary, and your sinusoid. Continuous capillaries, very they are continuous, meaning the cells, the endothelial of the endothelial layer, they are connected to each other. They are very tight. So talagang masikip ang masikip. They have they have very tight junctions connecting each other. Okay, you can see here there is an intercellular cleft between each of the cells, and then this cleft nakakonect yung mga cells tightly with well-developed occluding junctions. And these types of capillaries, they're found in muscle, connective tissue, lungs, your exocrine glands, and nervous tissue. Now, ultrastructural studies show numerous vesicles indicating transcytosis of macromolecules in both direction across the endothelial cytoplasm. So we've discussed transcytosis, or at least not we, but it have they have touched on transcytosis in the leg, on how material traverses from one cell to the other side of the cell, right? Substances like glucose, water, small hydrophobic molecules like gases and hormones, as well as various leukocytes as well. Here in this picture, you have your capillaries in a muscle tissue. Now, just a disclaimer, because because capillaries are already very small in structure, we will not be able to observe the or differentiate the three different types of capillaries under light microscope. Okay. Generally, we can only assume what type of capillary we will find based on the organ that we are observing. The second type of capillaries is your fenestrated capillaries, and they have a sieve-like structure that allows more extensive molecular exchange across the endothelium. What is a sieve? Just look up your sieve. It's a strainer class, but it's like a fine mesh. Your fine mesh sieve. Um, so it has many tiny holes dotting around the tunica intima. And these fenestrated capillaries, they are common in the small intestine, which is the primary site of nutrient absorption, as well as in the kidneys, which filter in the blood. So here in this picture below, it is a glomerulus, and it contains your glomerular capillaries. The third type of capillaries are your discontinuous capillaries or sinusoids. This is continuous because hindi magkadikit-dikit yung endothelium. In fact, they are almost separated. They have varying separations from each other, leaving large holes along the tunica intima of the uh, capillary vessel. Even the basement membrane is incomplete, allowing direct access for blood cells to exit and enter. Now, why would we want this type? Now, uh, your blood cells, for example, we don't want it going out of the blood vessel, like we've mentioned before, unless we want to, kasi class. Now, when would we want our red blood cells to exit the blood vessel? If, for example, we require them to be filtered, refurbished, or recycled, or sequestrated, or even for the entry of new blood cells. So we can find these sinusoids in organs like your liver, your spleen, endocrine organs, and of course your bone marrow. So here in this slide, I believe this is a spleen. Or is this liver? This is a spleen, and you can see erythrocytes being exiting out of the capillaries. After the blood reaches the capillaries, they have to exit the venules, um, and then they go back to the heart at this point. So now we discuss our veins. Your veins, this is a low pressure component of the blood circulatory system responsible for carrying blood from the capillary network back to the right atrium of the heart. The force impelling the blood towards the heart, often against gravity, is a combination of contraction of the smooth muscle by the vein wall and external compression of veins by contraction of skeletal muscle, especially in the specifically in the lower limbs. Your venous system also contains valves. So let's take a look at those structures and your different types of veins. There's three different types of veins. We'll start first exiting the capillary into a venule, which are the smallest veins. They gradually increase in size to form your small veins. And then your small and medium-sized veins, which include the external jugular vein, they have various sizes and structural features depending on their location. Now, similar again to the artery and capillaries, they also contain your three layers, your tunica intima, media, and adventitia. However, if we go back, I did not mention this part. Oh, where's that picture? The picture is missing. Okay, I don't have that picture anymore. Regardless, so ayan ang small and medium-sized vein. From your medium-sized veins, we will contain now your valves, your vein valves. And the job of the valve is to allow blood to, or prevent rather, to prevent blood from backflow or flowing backwards against, especially against gravity. So, for example, if you're standing up, your legs are pointing down. How does the blood from your feet go back up to your heart if there is limited amount of blood pressure? Your valves help with that. That way, it won't go back down to your feet. So here you have your valves that can be seen in your blood vessels, and they are folds which usually arise from the tunica intima as well. Then you have your large veins, which include your superior and inferior vena cava and your pulmonary veins. These veins possess cardiac muscle in the tunica adventitia for a short distance as they enter the heart. This layer also contains vasa vasorum and nervi vasorum because they are large. Although it is not commonly seen in veins, the internal elastic lamina may still be present in some large veins. Like here, as you can see, may elastic lamina siya. Here is the table differentiating your artery from your vein. Generally, when we say artery, it conducts blood away from the heart, regardless if it's oxygenated or not. Take, for example, your pulmonary artery, it's bringing oxygenated heart blood. It's wait no, it's bringing what am I saying? It's bringing the oxygenated blood away from the heart and into the lungs. While your pulmonary vein, it's bringing oxygenated blood from the lungs and into the heart. So artery and veins, they're more when we're talking about them, we're more talking about the direction of blood, if it's moving away from the heart or towards the heart. And the hindi porket oxygenated or deoxygenated. It could be too it can become too yes. Basta hindi deoxygenated or oxygenated ang basehan. It's the direction of where the blood is flowing. In terms of general appearance, the arteries are more rounder compared to your veins, which are irregularly shaped. Here you have an arteriole, very round. Here you have a venule, much larger, but more irregular. I think, do I have enough more pictures showing illustrating that? Here's a vein, more oblong, although your artery is also oblong here. Here, arteriole is much more circular. Your venule is now bending backwards, bending inwards. And then what else? The wall thickness of arteries are thicker than the vein, and in terms of valves, valves are present in your veins. Now, we proceed to your lymphatic vascular system, your lymph. So this will be discussed further in the LEC, but I will already start on this. just to uh yeah, just so you would know. So you um we have so many names throwing around: blood vascular system, cardiovascular system, circulatory system. Just to make things clearer, your circulatory system contains both your cardiovascular system, you know, the heart, artery, capillary, vein, and then your lymphatic vascular system, your lymph vessels and lymph nodes. And then the lymphatic system consists also of your lymphatic vascular system and your lymphoid system. But when we are talking about these two systems, more on the fluid balance and vascular system, and then the lymphoid system is more on the immunity side. And for today, we'll just focus on the lymphatic vascular and leave the lymphoid to the leg. So what first is lymph? How is it generating? So take a look at this here. Can I just oh, oh no. What happens?
What happens?
I can't tab. Hang on, I am appear to be having technical difficulties. Please hold. I'd like to draw here kasi. So here, you can see my screen. You have here your lymph. So how is lymph formed? Remember your blood, right? Blood enters the artery, passes through the metarterioles and into your capillary bed. Once they enter the capillary bed, fluid coming from the blood will, of course, extravasate into your peripheral tissues. And from your peripheral tissues, alongside this fluid will be your different nutrients and a bit of the dissolved gases, but mostly more nutrients, hormones, metabolites. They will all exit alongside the fluid here. So the fluid will have to travel throughout all of the tissues, and then in there, from the with the interstitial fluid, it will exchange nutrients with for waste products. The waste products will go into the interstitial fluid. The nutrients inside the interstitial fluid will go into the tissues. Afterwards, however, who is going to collect the fluid? It's not going to be the blood vessel wall. No. Um, the blood that's remaining, it has already removed some of its fluid, and maybe some of the white blood cells have also exited. The red blood cells should not have, but mostly the blood will return back into your venules. Your interstitial fluid, however, will have to return via your lymphatic capillaries. So, for example, pumunta siya dito, di ba? Pumunta sila sa tissues, and now they have to go to, in order for our body to reclaim this amount of fluid, they go back through your lymphatic capillaries. And this forms your lymph. So lymph is generally has the same composition of blood plasma. It contains all of the proteins, all of the hormones that's unused, of course, by the tissues, all of the metabolites unused by the tissues, and mostly the waste products. They're going to come back through your lymph capillaries. So if you take a look here, your lymphatic capillaries, they are blind-ended. What does blind-ended mean? Blind-ended. So here, ito, nag-end siya ng ganyan. This is a lymph capillary. Blind siya. Ending niya hanggang ganyan. Unlike your blood vessels, which, of course, let's get the red. Your blood vessels where, sure, it travels through the capillaries, but it will still go back to the peripheral blood system, like so. It's closed-ended. Your capillaries for your lymph, however, is blind-ended. So, and then here, papasok from the interstitial fluid back into the lymphatic capillaries. That's where how we reclaim our interstitial fluid as they traverse the tissues. If interstitial fluid cannot go back to the lymphatics, then of course that would leave interstitial fluid into our tissues in a phenomena called your edema. So that's the job of your lymphatic capillaries, okay? So now I hope na clarify na iba ang ang lymph, iba ang blood, okay? Okay. Um, some notable lymphatic capillaries are your lacteals, by the way. Lacteals are specialized lymphatic capillaries. So this won't be, again, the first time you've seen your small intestine. You have a hole here, which is found in the lamina propria of the mucosa of the small intestine. This is your lacteal. And the lacteal is a lymphatic capillary for the absorption, where your fats absorbed by the villi will move into and then eventually return into the blood. After your lymphatic capillaries in the tissues, of course, they will arrive in larger vessels called lymphatic vessels. And compared to your, of course, they also share the same shape and structure of your arteries and veins. They have a tunica intima, media, and adventitia. But one differentiating characteristic is that lymphatic vessels don't have blood cells. Rather, they don't have red blood cells, erythrocytes. What you would find instead in your lymphatic vessels are lymphocytes, because lymphocytes, they arise, and or rather they arise from bone marrow, but they develop in the thymus and the lymph nodes, where they are connected through the lymphatic vascular system. You could also see some coagulated plasma, but that's gonna be difficult to see. Either way, if you see either coagulated plasma or lymphocytes in a vessel and no blood cell, then you're most likely looking at a lymphatic vessel. As you can see in this picture here, you have a lymphatic vessel on the left and a vein on the right. Structurally speaking, they're quite similar, and it's difficult to differentiate them. The key factor in differentiating them is, of course, either the presence of absence of erythrocytes or presence of absence of only lymphocytes. Lymphocytes are not leukocytes. Um, Lymphocytes meaning the agranulocyte specifically, because these are your immune cells, which are developed they are produced in the bone marrow, and they develop in the thymus and in the lymphoid organs. More on that on the lecture. And then you have your lymphatic vessels. They also contain valves, and they also prevent backflow of fluid. Your lymphatic vessels all throughout the body, they will eventually all lead up back through either your thoracic duct and into uh, or your right lymphatic duct, which are quite near towards the veins here, your right subclavian and left subclavian vein, where they will dump all of that fluid into the blood, and then the blood will again redistribute it throughout the body. So ayan, that's how we balance. That is the purpose of fluid balance. It's how fluid that goes into the tissues come back into the blood system. They don't automatically get reabsorbed through the capillaries. They get reabsorbed first by lymphatic capillaries, and then into the lymphatic capillaries go to veins, and then into the ducts and into the larger blood veins. And then you have your lymphatic nodes and your diffuse lymphoid tissue. They are more on the lymphoid system now at this point, so we're going to leave those for the lecture. Hooray! We are done with our discussion for exercise 11.